So we're going to continue with our discussion on prokaryotic gene regulation. I can recall when I got to grad school being unsure really what I want to focus my research on, and it was within a course called Bacterial Gene Regulation that I really got excited about this topic. I think it makes so much sense. It's so simplistic in the approach, but yet so complex when you start to discuss the ability to turn on and off gene expression. So we've discussed the lac operon in class. We talked about the idea that there's a lac-I gene that encodes for an inhibitor protein, and that inhibitor protein is going to turn off gene expression when those enzymes involved in lactose metabolism are not required. And we're going to take a slightly different shift here. We're going to look at the lac operon. In this case, we're going to look at it being positively regulated, positively upregulated by the addition of an activator protein. So an activator protein is a protein that binds to a promoter region and it enhances transcription. We talked about this in class. It can do so by enhancing the binding constant, or it can do so by affecting the rate of isomerization or strand opening. So CAP stands for catabolite activator protein. It responds to changes in nutrient availability, and it does so by binding to a small molecule called cyclic AMP or CAMP. When catabolite activator protein, or as I'll call it from here on out, when CAP binds to CAMP, what you have is now an active form of the protein that is now able to upregulate the expression of genes that are required under certain metabolic conditions. So in the default state, the operons controlled by CAP is off when CAP's not bound. So what that means is when you have very low levels of cyclic AMP, you have very low levels of active CAP protein, and when that CAP protein's not active, the expression of that cluster of genes is off. Now, why would CAMP be repressed? Why would levels be low? It has to do with glucose, which I hope makes a lot of sense. We talked about glucose being the preferred substrate in glycolysis, etc., all involved in ATP production. So when glucose is around, those genes and those enzymes involved in lactose metabolism are not required. So when glucose is available, levels of cyclic AMP decrease, therefore preventing the activity of catabolite activator protein. Right, so think about from a cellular standpoint, you're trying to conserve resources. You don't want to express genes and translate the mRNAs when you don't need those enzymes and those proteins around. So now what we have is the ability to respond to nutrient availability through the use of small molecules. This is a concept we talked about a week ago on Friday the 21st. So now let's look a little bit closer at the CAP protein. CAP in its active state is a dimer. Now, one of the things I want to say is this. We still have a blog we have to keep this semester. We have a journal article that we have to add for Chapter 13. If you do a search for CAP, or in some places it's called CRP, you will find extensive studies have been done on this protein. Probably one of the most well-characterized proteins in all of biology. It's been studied from the genetic point of view. It's been studied from the biochemical point of view, the structural point of view. I mean, this protein has been dissected in many different ways, and it's really given us a nice picture of what a gene activator protein does. So here we have CAP as a dimer. Now, if I were to ask you what does it mean to be a dimer, what would you say? That's right, a dimer has two subunits. Now there's some symmetry here when you look at these two subunits. They are mirror images. So, for example, when you look at hemoglobin, hemoglobin has two alpha chains and two beta chains. We often call hemoglobin a tetramer. Here we have two identical peptide chains that if you look here, they actually join at a domain called the dimerization helix. it's this region of the chain that allows these two subunits to interact with one another. And when we form the dimer, that's going to be a very important part of regulating the process. And it's through this very long alpha helix in which that occurs. If I'm not mistaken, I believe there's many hydrophobic residues within this helix, and those hydrophobic residues are going to bury themselves. Now, another structural feature I want to point out is right here. Now, this is what we call a co-crystal. We have the protein that's been crystallized, the structure has been solved along with DNA. So DNA, the double helix is here in orange and blue. The protein is bound to it. In these regions, we have what's called a DNA binding motif. It's often called a helix turn helix motif. What a motif is, it's a common structural features among proteins that have similar functions. So for proteins that bind to DNA, many of them share this common helix turn helix motif. And what you can notice here is that protein is settled down nicely in the major groove of that DNA. The third important structural feature I want to point out is the small molecule cyclic AMP. We could see that binds in a domain called an effector domain. The effector domain is the part of the protein that binds cyclic AMP. So what you see here is in a protein you have three distinct functional regions. You have one region that's involved in binding to DNA. You have one region that's involved in forming this dimer. And then you have a third domain that's involved in binding the small effector molecule. And what you begin to find is that you see these common structural features in proteins that behave similarly. So CAP has been a great model protein when starting to examine other regulators within E. coli, which is where this protein comes from. So now, when can CAP do this? When does the helix turn helix domain, when is it in the proper conformation in order to bind? Well, this is where CAMP comes into play. When CAMP binds the CAP, now what we have is we have an active protein that is going to be able to bind to the DNA in and around the region of the lac promoter. If you recall, we talked about bacterial promoters in class. We mentioned that they have a -10 and -35 element. That's where RNA polymerase is going to bind. Here we have the +1 site, which is where transcription is going to begin. And then what you'll see is upstream you'll find the CAP binding sites. And you'll find them at these locations, -41, -61. You may even be able to go a little bit further downstream. But what you notice is that these sites are centered around 20 bases apart, which would put CAP on the same side of the DNA. Because recall, the DNA is twisted here. So what you would see is you'd see CAP bind in these regions. And what happens now, and you can see it in this crystal structure, and actually I'll erase the ink on the slide. When CAP binds to DNA, what it does is it promotes a bend. This crystal structures, you can see it's a 90-degree angle. And because it bends the DNA, it's that bending that brings RNA polymerase into the proximity and the conformation that's required in order to overcome limitations in either the binding constant, the rate constant, or both. And so when you overcome those kinetic barriers and those barriers in binding, what you're ultimately going to do is you're going to increase the rate at which transcription occurs. Now let's think big picture again. When glucose is high, that decreases the amount of cyclic AMP available in the cell. And when you decrease the amount of cyclic AMP, what is that going to do to the activity of CAP? Well, we just discussed this. Cyclic AMP is important in putting CAP in a conformation that allows it to bind to DNA. So when CAP is not around, what you have is you have an inactive form of cyclic AMP, of CAP. And when CAP is inactive, what you end up with is lowering or lower rates of transcription of the lac operon. Now I hope that makes sense, right? Because when glucose is around, there's no need to express those proteins involved in lactose metabolism. However, now when you release the cell from this glucose requirement, meaning when glucose is not available, what begins to happen is levels of cyclic AMP increase, which results in active CAP. When CAP is active, it can bind DNA, increasing the rate of transcription, and therefore what you end up with are the proteins involved, in this case in lactose metabolism. So here's a review of all the different inputs and all the different controls here. When both lactose and glucose are high, glucose is the preferred substrate, therefore the lac operon is shut off. The reason it's shut off, we just reviewed on the last slide, because cyclic AMP levels drop, CAP is not active, can't activate transcription, therefore a bacteria is only going to use a single substrate. And again, that makes sense. Use one substrate. When that substrate runs out, then you can switch substrates. Now, what would happen when lactose is high and glucose is low? Well, we have a few different inputs here. First of all, the lac operon's turned on because allolactose levels begin to rise, turning off the lac repressor. And when the lac repressor is off, it can't bind to operator. So now that's one point of control. Without repressor being bound, you can't turn off transcription. In addition, now, when cyclic AMP levels begin to rise, CAP is bound to the DNA. The DNA is bent at a 90-degree angle, and now the bacteria is going to synthesize the production of proteins that are involved in metabolizing lactose. Now, when lactose is low and glucose is in either state, whether it's high or low, the lac operon is shut off, and the reason is simple. Whether you have glucose or not, without the presence of lactose, that lac repressor is now in the on state, and when the lac repressor is in the on state, it's going to bind to its operator and essentially blocking those genes from being produced. So that's what we just talked about, how you can positively control a protein involved in catabolic reactions, right? The breakdown of something. Now I'm going to show you a little bit different view here. I'm going to show you the trip operon. Now the trip operon is a series of genes that encode for enzymes required to make the amino acid tryptophan. What kind of metabolic reactions make a product? That's right, anabolic reactions. So when a cell, a bacterial cell, lacks tryptophan, it has to make its own. Therefore, we're going to need to turn on that operon in the absence of trip in the cell. Now you notice, if I can compare quickly, that's very different than what we just discussed. Right? I wanted to turn on the lac operon when lactose was present. Now I want to turn on the operon when the substrate, in this case tryptophan, is absent. Very different. Now how can we regulate the expression of this operon? We have another repressor protein encoded by a gene called TRPR. Again, it got its name because it's the tryptophan repressor. We're going to see when the repressor is bound to its operator, transcription is inhibited. However, when tryptophan levels begin to drop in the cell, TRP repressor is no longer active, and now the genes involved in making the tryptophan for the bacteria are turned on. So therefore, if you think about the switch here, when tryptophan levels are high, tryptophan itself turns off the trip operon. When tryptophan levels are low, the repressor can no longer bind, therefore expression is turned up. So in this case, the product, tryptophan, the amino acid, acts as the signal molecule. And in this case, it acts as a repressor molecule. Let's take a look. Here's what we have in the case of low tryptophan. In the case of low tryptophan, I'm going to require the expression of these 5 genes whose ultimate jobs are to synthesize enzymes that are going to make tryptophan for the cell. So when tryptophan is low, the repressor can no longer bind. It's in its inactive state. When it's inactive, now RNA polymerase can bind to the promoter and now to synthesize a polycistronic mRNA, which ultimately is going to yield tryptophan for the bacteria. Now, what's going to happen when tryptophan's available to the cell through its environment, for example? Now, as that tryptophan then enters the cell, it's going to bind to the repressor. In this case, it's going to turn on the repressor. Now, is this similar or different to the example of the lac repressor? Actually, this is different if you think about it. In the case of the lac repressor, when there was high lactose, the repressor was off. Right? The allolactose acted as a small molecule that regulated the activity of that protein. But in this case, when tryptophan is high, the repressor is on. So you can see the difference between regulating catabolic genes and regulating anabolic genes. In the case of catabolic genes, what we do is we induce those operons. But in the case of anabolic genes, we repress those operons. So here, as tryptophan binds a repressor, it causes it to be in the active state. The repressor in the active state binds the operator just downstream of the promoter, thereby blocking expression. Now again, to me, a lot of common sense. When you don't have tryptophan, you need to make it. When you do have tryptophan, you don't need to make it. Conserve resources. Bacteria are constantly switching their gene expression in response to nutrient availability, because in order to survive, bacteria are constantly having to carry out cell division. And in order to carry out cell division, energy is required. So there's a summary of what we just talked about. Lac repressor binds to its operator in the absence of the small molecule. In this case, the repressor binds, the TRP repressor binds to the operator in the presence of that small molecule.